Going to see the major difference between these four rotifera: ENTOPROCTA, PHORONIDA, and ECTOPROCTA. So one by one we will compare all four. So the first thing is that I'm going to make a table. Okay, here we write rotifera, then the second one is our ENTOPROCTA, third one is PHORONIDA, and the fourth one is ECTOPROCTA. Okay, we'll compare all these in a very easy manner. So here are the features. Okay, so the first thing, that if the key structure will compare, then rotifera has corona. In their anterior part there is corona, while in ENTOPROCTA there is tentacular crown. In PHORONIDA there is LOPO4, and same in ECTOPROCTA also there is LOPO4. Okay, so on the basis of its structure, rotifera has corona, ENTOPROCTA has tentacular crown, PHORONIDA has LOPO4, and ECTOPROCTA has LOPO4. So remember this. Then the second feature through which we can differentiate them, so that is their feeding apparatus. So in case of feeding apparatus, the rotifera contains mastex. Mastex is a muscular pharynx-like structure, and they have trophy also. Trophy is like teeth of rotifera. Okay, so they are used for grinding and all. Then in ENTOPROCTA, for feeding apparatus there is ciliated tentacles only, through which they used to do feeding. Ciliated tentacles. Okay. And in PHORONIDA there is LOPO4 only, through which they used to do feeding, and here also LOPO4. Okay, so write that. Then the third thing through which we can differentiate them is their a part of anus. Okay, anus, where it is situated. So in rotifera, anus is situated in the posterior part. Okay, while in ENTOPROCTA, is the name indicating. Ento means it is inside the tentacle ring. So anus here is inside the tentacles ring. Okay, now here in PHORONIDA, this anus is in outside the LOPO4, outside. This LOPO4. Here also ecto means outside, so outside the LOPO4. Okay, now the next thing through which we will differentiate them is their body cavity. So body cavity, on the basis of body cavity, so in rotifera we see that there is pseudocoil. Okay, pseudocoel or pseudocoil, you will say pseudocoel. While in ENTOPROCTA it is reduced body cavity is found, reduced or uncertain. Okay, not known that much. While In Phoronida, they are completely coelomate, and they are also ECTOPROCTA are also coelomate. So we are saying that ECTOPROCTA and Phoronida are more similar to each other. Okay, coelomate. Now the next difference that we can see between them is excretion. How there is excretion among them. So on the basis of excretion, in ROTIFERA we find protonephridia, while in ENTOPROCTA we find this excretion is through general body surface, means by diffusion. Okay. Here in Phoronida we find metanephridia, more advanced metanephridia, while in ECTOPROCTA there is mainly diffusion, through diffusion only. So here we have to remember that there is diffusion, while in Phoronida they are more advanced. Here is metanephridia. Now through circulation. So how circulatory system in them, on the basis of circulatory system, how they are differ from each other. So circulation here is completely absent, here also absent. Okay, here in Phoronida we find closed circulatory system, and in ENTOPROCTA there is no specialized system, Okay, now other difference that through which we can differentiate them is the larva. So larva, it is found in only two of them. Larva, here we don't find larva, here also, but in Phoronida we find larva that is known as actinotroch larva. That is very important, actinotroch larva of Phoronida. Okay, so here we are writing Phoronida of actinotroch larva. Then ECTOPROCTA we find siphonoids, right here, siphonoids in many marine form. So here in marine form we can find this larva, siphonoids larva. Okay, then some special feature in them. There is special feature, that is mictic and amictic egg. We find here in case of ROTIFERA we find mictic and amictic egg. So mictic and amictic egg are nothing but they are like amictic egg, they at the end forms female, while in mictic egg we find there can be female as well as male. Amictic egg is actually a egg where we find lots of layers, so it is for unfavorable condition, and when favorable condition comes then they get converted into zygote and form a new organism. Okay, so amictic and mictic egg is a feature of ROTIFERA, while a special feature in ENTOPROCTA we can see is the calyx and stalk. Okay, calyx plus stalk, this feature is specially we find in ENTOPROCTA. Tube feet and hemoglobin we find in Phoronida. This is special feature of them. Tube and hemoglobin. So completely Phoronida are more advanced than any other. Okay, hemoglobin. While in ECTOPROCTA we find zooid and colony. Okay, zooids plus colony form. This is special feature of them. Now the next thing through which we can differentiate them is the reproduction, how reproduction take place in them. So in case of reproduction, okay, in case of reproduction, in rotifera we find parthenogenesis. So here mictic or amictic egg. So amictic egg goes through parthenogenesis only. So here parthenogenesis के through reproduction होता है. Parthenogenesis most common in rotifera, okay. And in entoprocta we find sexual reproduction. Sexual plus budding is also found in the form of asexual reproduction, okay. Phoronida completely sexual. This is important thing. Phoronida we find only sexual reproduction, while in ECTOPROCTA, एंटोप्रोक्टा और एक्टोप्रोक्टा दोनों में सेम रिप्रोडक्शन हो रहा है, सेक्सुअल प्लस बडिंग, okay. Now the next difference through which we can differentiate them is the examples. What are the examples देख लेते हैं. तो एग्जाम्पल्स में देखें तो रोटीफेरा में मोस्ट इम्पॉर्टेंट मेंबर दैट इज़ ब्रैंक्यूनस. ब्रैंक्यूनस इज़ एन इम्पॉर्टेंट मेंबर ऑफ रोटीफेरा. एक ही एक लिख लेते हैं. एंटोप्रोक्टा में देर इज़ लॉक्सोमेला. लॉक्सोमेला इज़ द इम्पॉर्टेंट मेंबर, while in case of Phoronida there is Phoronис, okay. Phoronis. So Phoronida is similar like Phoronис. So Phoronis is a member of Phoronida, while in the case of ECTOPROCTA we see there is two most important member, that is Bugula and Plumella, okay. So these two are the members. So these is the major difference between rotifera, ECTOPROCTA, ENTOPROCTA and Phoronida. Here we see that Phoronida and ENTOPROCTA are much similar to each other. Baki spatial functions we have seen. So this is all about comparison between them, okay. Thank you.